Good morning, everyone. I hope you're having a very good day. It's my pleasure to moderate this next session on next-generation antibody-drug conjugates, challenges and innovations in immunotherapy. It is my pleasure to also introduce my four esteemed panelists here. And I will get started with Dr. Bin Liu, who is a professor at UCSF, and I'll request Dr. Liu to introduce himself in a few words and his work in the ADC field.
Okay. Bin Liu, I'm a professor in Department of Anesthesiology, also member of the UCSF Helen Diller Comprehensive Cancer Center, also member of the Parker Institute. So our work is on new target identification by a non-gene expression-based approach. So to differentiate from the traditional way of target identification, we use antibody library-based direct protein-protein interaction to identify the novel epitopes on the tumor cell surface and arm them for ADC development. In the program of prostate cancer targeting, we have ADC in phase 2 trial, also a PET imaging study ongoing. I'm looking forward to the panel discussion on the next generation, including bispecific and the multimodality combination.
Thank you very much. Our next panelist is Dr. Hans-Peter Gerber, who's a Chief Scientific Officer at Sutro Biopharma, and I'd like to welcome him and request him to introduce and say a few words about his work in the ADC field.
Yeah, thank you. I'm happy to be here today to talk about ADCs. And so I started working on therapeutics 30 years ago, actually at Genentech around the corner. Worked there on Avastin for 11 years and then got fascinated by antibody-drug conjugates because they would do a thing a naked antibody like Avastin wouldn't do. You could regress already existing and established tumor with an ADC. You couldn't do that with an Avastin. You had to start early or in combination with chemotherapy. After 11 years with Avastin, I moved to Seattle Genetics then. I worked there for 3 years on ADCs. That's kind of the boot camp of ADC development. That's where a lot of things happened at the time. That was 2006. And then went over actually to the East Coast, joined the Pfizer team in Pearl River, actually at the site where the first ever developed ADC was being developed and approved. That was the CD33 Mylotarg with collegiamycin payload. We built up a rather sizable ADC group at Pfizer. We had developed about 8 ADCs, 2 INDs. Some of these are still going forward into clinical trial, and we got CD22 approved while I was there. And then moved on back to the Bay Area, where I was involved in several startup companies working on bispecific CD3, bispecific, but also TCR-T and CAR T cells. And then about 1.5 years, I joined Sutro. I actually founded an ADC company between that, and then I joined Sutro. And now leading their ADC R&D group, where we're going to talk about using a technology that is unique. It's expression in bacterial extracts, and that allows us to do things, engineering things with ADCs that conventional methods do not allow you. And we can talk about more about how that is in the benefit of the ADC development cancer patients at the end.
Wonderful. Thank you very much for joining this panel. Our next panelist is Dr. Stephen Eck, who's a Chief Medical Officer at Macrogenics.
Hey, it's great to be here. I'm a synthetic chemist turned hematologist oncologist by training. I spent most of my professional career on various aspects of drug development, particularly in oncology. My experience with ADCs started when I was attending at a physician at Penn on the bone marrow transplant service, and this was just a totally novel therapeutic when it came out, but it had a lot of difficulties, and I think it's interesting to see how we've gone from Mylotarg to now over a dozen approved ADC therapies. My main interest now is in the clinical development space. ADCs offer some really unique challenges, particularly on their pharmacology, how you combine them, and how we figure out how to integrate them into existing care, if not replace current standards of care.
Thank you very much, and welcome to the panel. And last but not the least, we have Dr. Amrita Kamath, who's a vice president in translational pharmacokinetics and pharmacodynamics at Genentech. Amrita, if you can just say a few words about your involvement with ADCs.
Yeah, thanks, Sandhya. So I've been at Genentech since 2006 for 19 years now, and started working in ADCs since then, and gone through, the early ADCs, MMAEs, and various different cytotoxics, and now working on the next gen ADCs. So it continues to excite me, but also challenge me continuously. So since I work in the preclinical area, so very focused on optimization of the molecule, and then how do we translate the PK/PD characteristics and figure out the therapeutic index and what models are translatable. So that's my main focus.
Wonderful. So as you can see, we have a panel that really goes all the way from target identification through IND submissions, as well as late-stage translational aspects, both preclinically as well as clinically. So really excited to engage all of you in some challenges and promises of next-generation antibody-drug conjugates. And for those of you who may not know me, I personally have also had an opportunity to work in the ADC field for over 2 decades, with an opportunity to work in the Kadcyla, Polivy, some of the VCMMA, SN38, and also the DM1 containing antibody-drug conjugates. So I think there's a lot of promises, but as we will be discussing, there are quite a few challenges as well. So for those in the audience who may not really know the basic structure of an antibody-drug conjugate or the construct of an antibody-drug conjugate, maybe I can request Dr. Gerber to start first, and then Dr. Liu to add on, what the basic construct is and also what the next generation of ADCs comprise.
Right, yeah, no, happy to do that description of the anatomy of an ADC. So there's 3 elements that are combined in one drug, and the first one is the antibody, and that is used as a vehicle to target tumor cells. So you're trying to have an antibody that targets the tumor-specific antigens to bring the compound to the tumor. And then you actually combine that antibody. There's naked antibody. When you block the function, you get enough activity to have a meaningful tumor response. But to enforce and empower these naked antibody, the natural thing is to conjugate something that will kill the tumor cells other than blocking the biological function. And cytotoxin have done that since 30, 40, 50 years. So the initial idea is then to conjugate via a linker, that's part number 2, the payload, that is part number 3, to the antibody so that the antibody finds the tumor, and then if you get the right target, it actually would internalize into the tumor and release that payload inside the tumor. So the benefit of that would be, and you can see that on all, it works wonderful on PowerPoint. The benefit of that is that you get a selective tumor delivery of chemotherapy, avoiding the systemic toxicity, of course. And then if you do this right and you get the right target, you get continuous delivery of chemotherapy into the tumor, maybe over 2 or 3 weeks, because usually you dose every 3 weeks. So you get this enhanced anti-tumor effect and the improved safety. So this is the base model. And now, of course, the antibody engineering has emerged. And there's components like we had this just in the previous panel, what happens if you target 2 antigens? Then can you overcome resistance? Can you cover more tumors? Or can you actually reduce the specificity more and you only target 2 antigens that are selectively expressed together on the tumor but never on normal cells? So that's one additional approach on top of the base model that has been pursued for over 20 years. We can have a round and how that is being pursued and the experience we all made with bispecifics. The next one there in that enhanced ADC category would be dual payloads or multiple payloads. And that is where I think is a significant benefit. And so I'm going from that, how can we make therapeutics better? One would be dual targets to overcome resistance. In the ADC field, we have the other choice. It's the payload, multiple payload, to overcome resistance mutations against one payload. And that seems to be now that we have multiple of these ADCs in the clinic, those patients who have sequential ADC treatment, sometimes ADCs that have the same linker payload, different companies, different targets. And what they see, if you get this an additional sequential treatment of a second ADC, once you relapse from the first ADC, you see resistance towards that ADC, and in particular, it's resistance towards the linker payload. If you then take a second ADC with a different linker payload, the patient will respond to the second as if they hadn't seen a previous first ADC. And so this is where I think is a huge opportunity for future engineering to make these dual payloads. And if you listen to the panel this morning, they said they all agreed on one consent information based on HIV research. If you have triple target, like this protease inhibitor that target 3 proteases in HIV, they could induce complete silencing of the virus. And they agreed that if you could target 3 targets on cancer, the same would happen. I didn't come up with that. That was the previous camel. There was consensus among the 3. Now, the next question would be if you then use, instead of 2, what we currently do, if you would actually conjugate 3 different cytotoxins, would that be enough for the cancer to run out of options to get mutations, resistant mutations to each of the 3 before you can eradicate the cancer? And I think that is a very attractive prospect based on science, what we just heard, to actually get us there and try this out. So
yeah, thank you. Thank you very much. I think you really bring up a very interesting point around the multiple payloads. And having worked in the ADC field, I know it is so hard to optimize the safety aspects with just one payload and one targeted antibody. So maybe I could call upon, some of our translational scientists here, Dr. Kamath, if you could comment on how do you envision having multiple payloads with multiple toxicities? How would we
Right. So
develop
when
it?
I was hearing that, right, it sounds very exciting, and we are excited about it. But so all these years we have worked with such limited payloads, right? The MMAEs, DM1s, galliumycin, and now we are talking about all these new different types of payloads with different, So just the understanding of even the one new payload is going to take a while for us to understand how things translate. And then putting 2 payloads, and let's say we are talking about a bispecific antibody, so 2 arms and then 2 payloads. And I just think it's of course not impossible, but it's going to be very, very challenging in terms of first manufacturing that molecule so that you get one product and not like, say, multiple products or... and then also, how do we optimize each part of it? So for example, even a single payload, what kind of DAR or drug antibody ratio you need, and then where do you conjugate it, which linker sites, and how will that impact pharmacokinetics and then safety? And as you know, right, just the drivers of, let's say, efficacy or safety are maybe the same, maybe different, and then for each safety endpoint, it may have a different driver of the exposure driver. So then how do we titrate that? So those are all the things that, I do think of a lot.
So as you can see, there's a lot of challenges in optimizing the construct of an antibody-drug conjugate with multiple payloads, multiple targets, and identifying a reasonable dose before we even put it in patients, looks like, right? And
right.
Maybe that's where we can have Stephen, if you could comment on how do you approach it, in the first human trials or clinical trials when you have these multiple complex modalities.
So we really have to solve for 3 or 4 things at the same time. So one is, as you pointed out, what you read on the PowerPoint slide isn't what the patient experienced. And a lot of you in the audience who take care of patients know this very well. So it's difficult to model a combination chemotherapy in experimental animals. FOLFOX6 didn't come out of a mouse model, right? Nor did CHOP or many other the combination therapies that are highly effective. So that's number one. So we may not have the preclinical proof that perfect combo we have in hand is actually correct. The second piece is the regulatory pathway. Do you want to start with mixing some ADCs together with different payloads and do that kind of trial? But then you have to have the financial resources and FDA regulatory processes to work with all those molecules. Or you just go for it all at once, make it, do your preclinical toxicology, and then try it in the clinic. Very different risk packages, very different costs and timelines to think about. And then the other part is the patient selection part. What can we know about the patient so we can a priori figure out who this combination, for whom this combination is going to work? And that patient selection part, I think, is a very difficult problem. We love to look at the surface expression of the target. Sometimes we're using archival tissue, and maybe that's not a very good representation of the patient's current experience. Is that target coming or going? We have great difficulty assessing a priori resistance to chemotherapy-like drugs. So those are all real challenges when you want to make these combinations. I'll give an example that we've recently experienced at Macrogenics, where we took a dual-specific antibody that blocks CTLA-4 and PD-1, that's lorigerlimab, and combined it with our Vobra Duo ADC, which targets B7H3 with a DNA alkylator. And so you've got 4 different biochemical processes in 2 drugs. And the complexity of running that trial, anticipating the ADCs and managing that is not what you see with your typical single agent cytotoxic agent. So that's what we're up against.
Wonderful, thank you. So I think we've seen reasonable success with the single payload and the first generation ADCs, and we just talked about some of the ADCs that the panel has worked on. But looks like, the second generation ADCs, which are more complex, is going to take a lot more understanding from a pharmacology as well as from the translational aspects to get it to a successful point. And with that, I also want to talk a little bit about some of the new mechanisms of action and also the immunostimulatory antibody drug conjugates, which are quite popular as well, in terms of at least early development stages. So maybe, Dr. Liu, I could have you chime in on your experience there.
Sure. After that, I want to comment a little bit on the bispecific and the dual conjugates. So generally speaking, you can use ADC to achieve immune stimulation by 3 mechanisms. One is very straightforward. PD-L1 ADC have been tried in phase 1 trials, so that directly activates the immune system. The second mechanism, people have conjugated immune activators such as STING agonists directly to the antibody, the EGFR, HER2, and the CCR2 by Takeda. Those STING agonists can stimulate interferon and the cytokine release. The third one, which is our own experience, so some ADC can in itself stimulate the immune system. So for example, in our CD46 ADC trial, we're trying to look for correlative with the response. We didn't see IHC correlate. We're not sure PET imaging correlates. But what we did see is that patients who responded to CD46 ADC have a reduction of myeloid cells and the concurrent increase of CD8 cells. So that means the ADC itself, in addition to directly kill the tumor, had an immunological impact, and that impact correlates with the response. Coming back to the bispecific and the dual conjugates, a couple of comments. I think Dr. Gerber summarized very well the bispecific can overcome heterogeneity in target expression and antigen escape. I just want to add that in addition to that, 2 more things you may pay attention to. First is bispecific. You can weaken the arm to make it, each arm make it moderate so that it can help tissue penetration. It can walk into the tumor tissue more easily than a high affinity monoclonal antibody. The second part is that by tethering the 2 antibodies together, the behavior of the antigen is no longer intrinsic to itself. So you can convert a non-internal antibody into an internal antibody just because you have a tethering effect of the bispecific. So that further enhance delivery. On the multimodality side, dual modality side, part of the thing, I'm just speaking for stuff we're doing in the lab, in terms of homogeneity and the characterization of double payload, we pursue site-specific conjugation. So some go on to the glycan, the asparagine 297. Some go on to through the cysteine bridge, go on to the cysteine residue. So you can achieve defined ratio 2:1 or 4:1. But nonetheless, I agree the characterization is a challenging. That being said, what I'm thinking is that we can broaden the concept of multimodality a little bit. So instead of thinking just two chemotherapeutic together, you can imagine the A could be a chemotherapeutic agent, the B could be a pathway inhibitor, such as tyrosine kinase inhibitor. Stretching this out a little bit, so in the lab, we're working on TCR conjugated with drug. So it's a TCR, T-cell engager ADC, not TCR, TCE ADC. So I have independent mechanisms that bring the T-cell function into the tumor killing. Also, we have a paper that's going to come out that we combine radionuclide, in this case, alpha particle radiation or beta. I mean, so lutetium-177 is good too. We use actinium-225 combined with our MMAE-based ADC. We definitely see enhancement, the single agent double labeled over the either radio labeled RIT or MMAE-based ADC. Now, thinking a little bit further, if you allow me, we have been thinking about tumor targeting by biological or chemical means. But ADC, one of the primary challenges is delivery. So we're exploring the concept of utilize some physical means, such as high-intensity focused ultrasound, so that those can be helpful in certain type of tumor to loosen up the tissue. Also through sound operation, disrupt the cell membrane. And the ultrasound itself have an immunostimulatory effect. So in itself, it's a good combination with ADC.
Wow, really very impressive. Talking about new mechanisms of action, these are really far-fetched and really excited to hear. Would anybody else like to add?
Can I just add, that was
comments?
really, actually really exciting. So, in addition to the dual payloads, what you said about using the bispecific to maybe increase internalization, so then you can get more of the, whether it's one payload or two, more of the payload in the tumor cell. Because what we are dealing with is, when you deal with these antibody-based conjugates, you will always have some level of non-specific uptake. But even if it's targeted, it's not always 100% targeted. You have some percentage going in your normal tissues. So one area we are, hopefully the field is looking at is how do you decrease that non-specific uptake, and then so that way we can maybe increase the dose that we can give, and then drive more of the molecule to the tumor cell. Because right now, for example, in like MMA payloads, you are limited, doesn't matter what antibody you use, you're limited at, all the doses are the same, around 1.8 milligram per kg. So I feel we are limited, we could get additional efficacy if we could break that barrier. And how do we break that barrier? So those are also some things, I hope there's advances in the field.
Yeah, I think this issue around being taken up non-specifically in a third compartment, non-tumor compartment, it's not in the plasma, it's not in the tumor, it's someplace else, is a real issue for many ADCs. We certainly saw this with vobramitamab, B7H3 directed ADC. And BMS also saw it in a paper they published in a CD70 directed ADC with the same payload. And you get these delayed pleural and pericardial effusions, delayed by many months. And this is not an on-tumor effect. It's not, but it's the release of the toxin from someplace else back into systemic circulation. And that's why you see persistent levels of the toxin, which is normally readily cleared, it persists because it's constantly being released. So I think solving that problem alone would be a major advance, along with actually figuring out some of the unique toxicities that we see with ADCs that we don't see with conventional therapies.
Very interesting. Yeah.
Yeah, I think this is just to put this a little bit in perspective, if we look back 20 years of ADC development, it started out by the entire field, Seagen and others, looking for more potent payloads. And the reason for that was that the first generation with doxorubicin failed because the payload was not potent enough. So there was this intense research focus on more potent payload, DNA, double-strand breaker, and others like collegiamycin, picomolar potency. And the winner, so just remind everybody, 5 years ago, ADCs were not the winning modality in oncology. It was almost a dying species. The breakthrough came with a new payload. It's the topo-1, the exatecans. And what this payload did, this is actually a low to medium potency molecule compared to the MMAs and the others. So why all of a sudden is a lower potency making a better ADC, a lower potency payload? And what it is, it's actually very simple. We all know that in ADC development, if you were to test these molecules for safety, and the problem is I had to go all the way to cyno to figure that out. You will find out that you can now dose these ADCs with okay potency, DOR8, you get almost like a VCMME potency, but you can dose at 30, 40 mg per kg. And these MMEs, the previous version, the potent one, you could dose at 6 mg per kg. So whoa, now you got 5 times more exposure. What does that do to an ADC? We always knew for ADCs, more exposure, better anti-tumor efficacy. There's nothing magic about that. But the lessons learned that a higher potency was not getting you to the point where you want to go, but you went to a lower potency now. Why is that? Because what he just mentioned, that off-target toxicity in tissues we don't understand, when these payloads get to those tissues, they do less damage. So in my thinking now, the big next breakthrough in ADC will be on payloads that do even more of that topo-1. So we should screen for the ones that do not harm normal tissues and have okay potency on the tumors, because we know we get with a DXD conjugate, we can have very good responses. We don't need to tune up the potency. What we have to make sure when it gets to the other tissues, and of course could be a 1-hour talk how it gets to the other tissue and what the mechanisms are and how we can reduce that, but that's not the point here. But it's just like the general concept, I think, when we go forward, finding of these more tolerable payloads and maybe adding 1 or 2 or 3 to an ADC, which is the stretch goal, of course. This is not easy. But that's kind of the way to go with the modality. And then the one thing also, we talked about target, how important they are. If you just conceptually, if this holds on, these emerging clinical trends, that once you get a topo-1 ADC, you no longer respond to a second one. It really tells you that maybe having 2 payloads to overcome that, we have to adjust to the emerging clinical trends. And so I think more payloads, different payloads on the same ADC may bring us to better benefits and ultimately better benefit for each patient getting these ADCs.
Yeah. I think you bring up very interesting points, but it also kind of brings up some of the additional challenge, right? Because even as you pointed out, we know if you can get a higher exposure, we can optimize better outcomes for patients. Having said that, with more payloads, more overlapping toxicities, or more tolerability aspects, how do you think about optimizing it for individual patients, and how can we improve outcomes for patients? Maybe I can open it up for...
Well, I think some of this is very empiric, and the simplistic answer is you're not going to figure it out before you get it approved. And just get it approved and then work out why it worked later, because that's often the case. You don't have enough N in your clinical trials with an effective drug to really know everything about it. So building in more translational medicine into your ADC studies will help if you can ask the right question, but sometimes we don't know the question that we should be answering. So I think that's step one. The other piece is the patient selection within indications. And I think that's still very difficult. It's clear with the tyrosine kinase populations, we know how to do that. You can screen for EGFR, so forth, mutations and so forth. I think for ADCs, we're still very rudimentary in how we decide who should get our drug. And it's still very much trial and error. And I think that's part of the problem.
So Amrita, do you see yourself conducting different kinds of experiments to answer some of these questions from a translational standpoint before we even get into patients?
You know, as Dr. Ik mentioned before, the preclinical models, at least for efficacy, even for chemo, we don't have that translation that well. So it's going to be really difficult to have a good translation for efficacy. I mean, in mouse, everything works.
Yeah.
And also maybe the tumor penetration may be much easier because in humans, it's much more difficult. So that tumor, when we make high affinity antibodies, we don't get it right into the tumor. So all those aspects makes it difficult, whether it's one payload or multiple payloads.
Yeah. Dr. Gerber, would you like to add to that?
Yeah, so I think the path forward to actually find what is the right mixture of these payloads and that are potent but not too toxic. I think we have very good models in hand and very validated model for ADC development. We wouldn't have that for bispecific to the extent or CAR-Ts. We don't have that, but for ADC we have those. For the efficacy, I would tend, and I think that's shared by AstraZeneca, by Pooja Chopra, that PDX models, when done right, give you a very good insight about what to expect in terms of pharmacology, what dose you need to apply to get tumor regressions or objective response rates in the right range. The safety one, we trust, I mean, based on the experience that we've done more than 10 cyno safety studies, what you find and what you see in cynos usually translates into humans if you do the right PK adjustment, with one exception. Peripheral neuropathy you cannot measure in cyno, but all the others you see if you know where to look for. So now we have the true foundation of data. You use PK modeling, you calculate what that translates into human, and you can actually estimate whether the TI of a dual is now compromised by one of the payload or not, and how you move that payload and what's the right ratio of the second payload is, you still get a better TI. Because we all know when you now, and actually we just designed these cynotox studies this week when having the tubulin payloads, which is the most successful class of payloads in ADCs. Over 50% of ADCs approved have tubulin inhibitors with topo-1s. And these are currently the leading ADC platforms. So in mixing those two, we all know that you want to look at safety because the tubulins, they have a more safety signal at lower dose. So if you add one of those, you then compromise the dose you can apply to topo-1. And getting out that mixture to the right level, did you have better efficacy and eventually equal toxicity or maybe a compromise? And what is that compromise you need to do? And what's the result in pharmacology? That is exciting, and that's going on right now. And we have the right tools actually to measure that preclinically and then go to the clinic. And then is the moment of the truth. I agree. There's nothing else in the clinic that tells you whether what you built is actually doing what you think it does.
Right, and I think I'll probably just bring up one more controversial question, which I don't think most people really like, is the Project Optimus that FDA is imposing upon all the oncology drug development. How do you feel about it for some complex modality like ADC, where we have to learn with patients and have to optimize it in patients? And
well, I think the way I think about it is a lot because it's a very difficult topic. I mean, I think Project Optimus is well intended, and I think the FDA has done a good job of pushing forward with that with appropriate caveats here and there, because we haven't done a good job historically about optimizing dose in a clinical setting. And you could look at something as simple as irinotecan when it came to market. Nobody used it. Less than 5% of the patients who got irinotecan got it at the labeled dose, right? And so the label should reflect the reality. So I think Project Optimus is well thought out. But immunotherapies and ADCs, I think, are unique or perhaps much more difficult to optimize. For instance, with the ricamycin payload, the dose response is flat. You get to a point where it works, and then above that, you're not buying much more as you go up. But finding that plateau is tricky. And so you're going to have to get some exposure, you're going to have to do some modeling, look at your free payload, look at your free toxin, and then find what we call your no-regret dose. Is it the perfect dose? No, but we're not going necessarily forward with the MTD, so to speak. That's a little tricky to do on the animal side. You need a good human data. You need more PK later in your studies. We tend to do a lot of non-compartmental PK in cycle 1. You get a bunch of samples. You get your nice distribution curve. But we need to do PK later because people are going to be on these drugs not for 3 months, but maybe for a year if they're successful. And this could be, very given more chronically than we're expecting. So I think you need to incorporate a Project Optimus. You may need to make a good faith effort to find a reasoned dose, not the MTD necessarily. And I think some of this is going to be worked out post-marketing. You're just going to have to keep beavering away at it until you can fine-tune it, which is how all chemotherapy got developed.
I know we have so much more to talk about it. There's just such a complex field and a lot of promise, but I know we are really running out of time. So maybe I'll have each of you, just starting with Amrita, in 30 seconds or less, what do you hope for, as we continue to work on these next generation ADCs?
Hope we can solve these problems. And I'm actually really excited about all these new formats and approaches that we're trying. And I'm also excited, we are now moving beyond oncology to non-oncology areas and just trying to see, how ADCs can be used over there to improve efficacy.
Wonderful.
I think we've covered a lot of the places we're going. I think one thing we haven't touched on is using antibodies to deliver more conventional, not so toxic drugs. Could you deliver enzalutamide with an antibody and decrease fatigue? Maybe it doesn't work better, but not having fatigue in prostate cancer would be a big accomplishment for patients. So I think we're going to find other applications where we can change the pharmacology of existing drugs by putting them on antibodies.
Thank you.
Yeah, well, I look at the two areas, the oncology, the near-term goals, where we can go based on clinical data, the learnings from the past. And I think the goal would be to have not only the exotecans, but something in addition that we can combine to further advance ADC technology, both on the safety and efficacy side. And then outside of oncology, of course, there's a lot of talking about, why don't we use ADCs for autoimmune disease? Can you target certain subset of T cells, maybe combined with TCR biology, and all these things are up there that I think that's really the 5 to 10 year horizon. Where can we have the most impact of targeting certain immune cells with ADCs or targeting certain inflammatory cells during inflammation or even bacterial infection, things like that. But that's really the future. I know there's some very focused biotech working on that, but it shouldn't stop with oncology. We should learn and apply to other more chronic diseases.
Makes a lot of sense. Dr. Liu?
So I'm struck by how little ADC injected into the human body actually got taken up by the tumor. Normally less than 1%. So 99% of the drug you injected just circulating in your body, go to liver, causing toxicities. How do we improve that? So ADC potential, I think, hasn't been fully realized. The barrier, one of the barriers is just the delivery itself. I'm not giving up on biological approach. The bispecific is a good approach, but in addition, I really think you need to integrate some of the physical approach, the ultrasound-based or even alternating electric field right now. The second thing I think, from my observation, the ADCs are going to succeed in the clinic probably going to involve the immune mechanism. It's not just the tumor debulking tool. It has to come along with some additional mechanism. And that lends the door open to either the ADC having intrinsic activity, or you can conjugate immune activator to this ADC, make it a dual payload, but the payload is not another chemo, but it's an immune agonist.
Thank you. Thank you very much. We do have a minute or two if anybody in the audience has a question or two to ask. We have one here and one here.
Sorry, I have a... Thank you. Lia Tziperidu, I'm an oncologist at MD Anderson in new drug development. So I have used many ADCs while we bring these trials, and the two key areas that I see that there is a lot of room for improvement are, one, developing prognostic markers for toxicity, because once we have significant improvement, but then many patients complain of a lot of fatigue, as mentioned with prostate cancer. And so it would be nice to know, and we have to stop treatment, and perhaps some of them cannot continue any treatment because of the severe fatigue. The other area is how do we optimize how we can have some patients to be cured with ADCs. Because in the era of immunotherapy alone, I have patients in my clinic who have been cured with some immunotherapies. But I have not seen, perhaps because these patients have very advanced cancer, cured with ADCs so far. So I was wondering, and I have not seen any combination of ADCs with PD-L1 inhibitors, and I go back to the comment of the last comment we had. So is there any, how can we, what are the mechanisms of resistance we need to overcome? And how can we move from what we have accomplished so far with ADCs to cure? Do we need to combine them in addition with other drugs? For instance, is there any combination with immunotherapies of ADCs?
There are studies ongoing in combination of ADCs with PD-1 and PD-L1 inhibitors. I don't think they have read out, but maybe Dr. Eck?
Yeah, so we did a study with our lorigerlimab, which is the bispecific CTLA-4 PD-1 inhibitor, in combination with Vobra. We haven't reported those data out yet, but that's a phase 1 combination that's designed to give you the benefit of the chemotherapeutic effect of the ADC and the immunostimulatory effect of the two checkpoint inhibitors. So you get three modalities in one.
Yeah.
I think the 19.4 with PD-1 also in ongoing clinical trials.
Yeah. We are out of time. I know we barely scratched the surface of this very complex and interesting topic, but I really want to thank my panelists for engaging us in a very interesting discussion, and thank you all for your participation. Thank you.